Peripheral Nervous System Highlight screencast will introduce neurons and neurohistology. The screencast is intended to provide background information for appreciating spinal nerves, cranial nerves, and the autonomic nervous system, which together comprise the peripheral nervous system. Neurons and glial cells are the parenchymal cells of the nervous system. Neurons are the functional cells, and glial cells are the supporting cells. The glial cells of the peripheral nervous system are called neurolemmocytes, or often just lemmocytes. Neurons specialize in excitability. They become excited, they conduct excitation from one end of the neuron to the other, and they transmit excitation to other neurons or to muscles and gland cells. Excitable cells have a polarized plasma membrane. The membrane acts as a barrier that keeps cations and anions separated. Excitation occurs when the barrier properties of the membrane decreases, so ions can flow through and neutralize one another or even reverse the polarity. In addition to this electrical gradient, ion flow across the membrane is driven by concentration gradients. In fact, concentration drives the ionic flow across the plasma membrane that produces the polarized plasma membrane in the first place. Thus, excitable cells are sensitive to local ion concentrations, which have to be at a certain level in order for the neurons to function properly. To protect the ionic environment, nerve fascicles are enveloped by epithelioid cells that establish a protective environment for axon conduction to take place. Most neurons are multipolar. Multipolar neurons receive excitation from synapses. They conduct excitation along the axon, and they transmit excitation by releasing neurotransmitters at synapses. The neurons are multipolar because the spherical cell body plays a significant role in integrating the total synaptic input to the cell. The other neuron type is unipolar. Unipolar neurons are sensory neurons. Sensory neurons become excited by peripheral receptors, such as mechanoreceptors or thermoreceptors or nociceptors that are excited by noxious stimuli. The cell body does not have an excitability role, so it is located along the axon in a ganglion near the central nervous system where it can be protected by bone. The terminal branches of a unipolar neuron are within the central nervous system where they excite the next neuron in the pathway. This is a schematic of a multipolar neuron. Around the cell body we have these poles. Each pole gives rise to a dendrite, except in one case the pole is an axon hillock that gives rise to an axon. The cell body plays an important role in integrating this total synaptic input. When the excitation reaches a critical level, it triggers an action potential which conducts excitation from one end of the axon to the other, where terminal branches synapse on the next neuron in the pathway. In the case of urgent information, conduction can be accelerated by wrapping the axon in myelin produced by neurolemmocytes. So the action potential jumps from node to node, skipping the internode regions. This is a transection of the spinal cord, which is central nervous system. Neuron cell bodies are located in the gray matter. You can see some of the large neuron cell bodies just barely visible at this magnification. Virtually all neurons in the central nervous system are multipolar neurons. This microscopic image shows a multipolar neuron with a silver stain. These poles are probably dendrites. There are synapses on the dendrites and on the cell body. These little dark circles are synapses. Thus, in a multipolar neuron, synapses are on the dendrites and on the cell body. This dissection image shows a cervical thoracic ganglion and a middle cervical ganglion. These are autonomic ganglia, and they contain postganglionic cell bodies that receive synapses from preganglionic cell bodies. Thus, the cell bodies in these ganglia are multipolar. This is an image of a microscopic section of an autonomic ganglia, and it shows multipolar postganglionic cell bodies. The red arrows point to satellite cells formed by neurolemmocytes. This diagram contrasts the multipolar neuron and a unipolar neuron. The axons are shown in red. The axons conduct excitation from the excitation input region of a neuron to the excitation output end of the neuron. In the case of the multipolar neuron, the cell body is within the excitation input region. The spherical cell body, in fact, plays an important physiological role in integrating the total synaptic input and determining when an action potential will be triggered. For the unipolar neuron, the cell body is along the axon. It is located in a spinal ganglion. Excitation begins at a receptor, in this case free nerve endings are sketched, and the axon conducts that excitation to the central nervous system. In the case of the ear, sensory neurons are bipolar. The excitation travels along the axon, including the cell body, and then it is released in the central nervous system. Sometimes the unipolar neurons are referred to as pseudo-unipolar because embryologically they are bipolar and then they transform to the unipolar state. This cartoon shows various receptors associated with unipolar neurons. The receptors are out at the peripheral branches of the unipolar neurons. Here are free nerve endings that come up from the dermis and branch in the base of the epidermis. These are encapsulated receptors. This is a mechanoreceptor. Several axons are branching among modified supporting cells. Here the supporting cells have formed an onion-type arrangement around a single axon. This is a tendon organ that signals muscle tension. This cartoon shows the spinal cord within the vertebral canal. This spinal segment has given rise bilaterally to a pair of spinal nerves. Each spinal nerve is attached to the spinal cord by a dorsal root and a ventral root. A spinal ganglion, which contains unipolar neuron cell bodies, is located on the dorsal root. This image is of a microscopic section through a spinal ganglion. The cell bodies are unipolar, so only one process is coming off them. Satellite neurolemmocytes surround the unipolar cell bodies, as indicated by the white arrows. When you compare the size of these cell bodies compared to the size of axons, you can see why the cell bodies produce such an enlargement of the nerve. The enlargement in the peripheral nervous system is called a ganglion. Components of the peripheral nervous system are either a nerve, a root, or a ganglion, and there are two types of ganglion. A nerve, which is what is typically dissected in the peripheral nervous system, consists of thousands or hundreds of axons, all enclosed by glial supporting cells and surrounded by connective tissue, which is called epineurium, perineurium, and endoneurium. This cartoon illustrates components of a peripheral nerve. It shows an afferent axon, which has receptors out at the periphery, and an efferent axon that's innervating skeletal muscle. The axons are ensheathed by myelin, and the myelin is produced by neurolemmocytes. Myelin is provided when nerves are carrying urgent information because it speeds up conduction. The action potential jumps from node to node. Bundles of axons are grouped into fascicles. Each fascicle will eventually become a branch of the nerve. The connective tissue associated with the nerve is epineurium, which surrounds all of the fascicles, perineurium, which surrounds an individual fascicle, and endoneurium, which is scant within a fascicle. The purple represents perineural epithelioid cells, which form a barrier that protects the ionic environment within a fascicle. The proper ionic environment is important for nerve conduction. This is an image of a microscopic section through an equine nerve. Epineurium is surrounding all of the fascicles. Each fascicle is surrounded by a perineurium. Inside the perineurium is a perineurial epithelioid layer with endoneurium and the individual axon not very obvious at this magnification. This is an enlarged image of a nerve fascicle. The perineural connective tissue is out here. This is layers of perineural epithelioid cells. The endoneurium is scant. Myelinated axons are prominent. Here's a myelinated axon. Here's the axon. This is the myelin around it. Here's the axon. This is the myelin. Processing of the tissue disrupts the myelin. Non-myelinated axons are also visible. These tiny dots, these clusters of dots then, represent individual non-myelinated axons. Non-myelinated axons are much more economical in the sense that they take up less space and they are used for less urgent information. They conduct much more slowly than the larger myelinated axons. This image shows nerve fibers in longitudinal section. This is a lower magnification and a higher magnification down below. The black arrows point to the axons. The material surrounding the axons is myelin sheath, and the blue staining material is endoneurium. This is a myelin node. Above we can see multiple examples of nodes. So this concludes our peripheral nervous system highlight screencast concerning neurons and neurohistology.